Good morning, students. In the last class we have discussed about cell wall. Now let us discuss about nucleus. The term nucleus was given by Robert Brown. We have discussed about preparing temporary mount of onion peel. Temporary mount of onion peel. So in this experiment, what we have done, we have taken an onion and one leaf. Many onion leaves will be there. So if you take the onion like this, there are many leaves like this. We will take the outer leaf, any one leaf. In this, if you break the onion leaf into two pieces and carefully lift it apart, this peel, if you carefully lift it apart, you will get that peel. So like this, so one small peel you will get. What will you do with that? You will be just putting it in water and then you are keeping it on a glass slide and you add a stain called saffronin. What is stain? Coloring agent. And you observe it under microscope. Then what are you going to watch? You are going to watch the cells, hexagonal shaped cells. You are going to watch. Right. So this is what you are going to watch under microscope. If you carry out the experiment in the laboratory, you are going to get the result like this only. Why it is round? Because the objective lens through which we watch will be in circular shape, so we'll get this type of thing only. So we can use saffronin or iodine solution, something. The entire peel, is it getting evenly stained? No. The central part, it is getting more colored, and remaining part it will be getting pale colored, right? Why they get differently colored? why different parts of cell get differently colored. Different parts of cells do have different structures. Different structures do have different chemical composition. So, different parts of cell get differently colored because of their differences in chemical composition. So different parts of cell have different structures that are having differences in their chemical composition, and due to the differences in their chemical composition, they get differently colored. They do not get evenly colored. So for staining, we can use iodine solution, or we can use saffronin, or we can use methylene blue, etc. Right? Whichever the coloring agent is available to us, we can use that particular coloring agent. So, in the previous experiment, we have observed the temporary amount of onion peel. So, onions we have to get from the market, and we have to do all these things. Instead of that, we can use our own cheek cells. So, how can we do mounting of our own cheek cells? Mounting of human cheek cells. Mounting of human cheek cells. First, take a slide with a drop of water on it. Take a slide with a drop of water on it. Then what do we do? We have to take an ice cream spoon or some mouth pick, or in our lab a particular instrument is available called scapula. Ice cream spoon or toothpick or scapula. So ice cream spoon, you know the shape. It will be like this. Toothpick, like this we will be having it. Scapula, almost it will be of in the same shape, but little lengthier. That's it. So any of these things you can take, and you can gently scrape the inside of your cheek. Okay, gently scrape the inside surface of the cheek. Inside surface of cheek. Using any of these things, you can gently scrape. And when you scrape, a white color material will stick onto the end, whichever you are putting inside. Now, whatever that it is getting sticked to any of the things, using a needle, you can transfer this thing to this slide. Okay, and then you can spread it evenly. And add methylene blue. It is also a coloring agent like iodine solution or saffron, and leave it for few minutes. Then observe it under microscope. What are you going to observe under microscope? You are going to observe the cells like this. Actually, since you are going to use methylene blue, they are going to appear blue in color, but I am drawing them on with the black color. Okay. All of the cells will be of same. They are of same shape because they are belonging to same structure, right? And inside the cells, a darkly colored spherical structure you can see. That will be almost near to the center of the cell. The darkly colored spherical structure that is found almost near to the center of the cell is called nucleus. The darkly stained spherical structure which is present near to the center of the cell is called nucleus. So this is how we are going to do the two activities: temporary mount of onion peel and mounting the human cheek cells to observe the nucleus. So these structures, the central spherical structure that is found in human cheek cells, it is almost similar to the darkly stained central structure that is found in the onion peel cells also. So what is nucleus? The darkly stained spherical, darkly stained central spherical structure of a cell. Central spherical structure of a cell is known as nucleus, right? This nucleus, if you look at the structure of the nucleus, how it is going to appear. The nucleus is bound by a double layered membrane. Assume that it is the nucleus which is bound by the double layered membrane, and these two layers, they invaginate at certain points. Invaginate means moving inside. They will invaginate at certain points. So small pores are formed these are called nuclear pores. This is nuclear membrane, and these are nuclear pores. How nuclear pores are formed? By the invagination of the outer layers of nuclear membrane. And these nuclear pores allow the transfer of material from inside the nucleus to outside, as well as from outside the nucleus to inside. Okay. What is the role of nuclear pores? They allow the transfer of the material from inside the nucleus to its outside, that is to the cytoplasm. The cytoplasm. And the nucleus contains genetic material in it. Genetic material is present in the nucleus. The nucleus is going to contain genetic material. This genetic material, it is present in two different forms based on the phase of the cell. The nuclear genetic material is present in two different forms based on the phase of the cell. A cell can be in two phases, either in dividing phase or non-dividing phase or condition, anything you can write. Dividing phase and non-dividing phase. During dividing phase, the genetic material is present in the form of chromosomes. During the non-dividing phase, the genetic material is present in the form of chromatin. Right? So genetic material is going to be present in chromosomes and in chromatin. Chromosomes form is seen in dividing phase of the cell. Chromatin form can be seen in non-dividing phase of the cell. What are the differences between chromosomes and chromatin? Chromosomes are rod-shaped structures. Rod-shaped condensed structures. Right? Means when we see them under the microscope, they are seen as rod-shaped condensed structures. And when are they going to be seen? Only when the cell is about to divide. When cell is about to divide, right? The genetic material that is responsible or that contains information for the inheritance of features from parents to the offspring or to the next generation is present in the form of DNA in chromosomes. Okay, so chromosomes contain the genetic material in the form of DNA. What is DNA? Deoxyribonucleic acid. Deoxyribonucleic acid. This, what is its significance? It contains the information for the inheritance of features, from parents to offspring or next generation, you can say. So totally what chromosomes contain? Only DNA? No. Chromosomes contain both DNA and proteins. Chromosomes contain DNA and proteins. The proteins present in chromosomes are called histones. If you want, you can remember, right? Chromosomes are composed of DNA and protein. And as I said, the information for the inheritance of features from parents to offspring is present in the form of DNA. And DNA molecules, they contain the information which is necessary for constructing and organizing the cells. DNA molecules not only contain the information for the inheritance of features from parents to offspring, but also they contain the information for the construction and organization of the cell. They contain, DNA molecules, they contain the information for the construction and organization of the cells. And each functional segment of DNA is called a gene. Each functional segment of DNA is called a gene. What is gene? Functional segment. Functional segment of DNA. Functional segment of DNA is called gene. If you take the cell which is not dividing, in that cell the DNA is present as part of chromatin material. The genetic material is going to be present in chromosomes in the cell which is about to divide, whereas the genetic material or DNA is going to be present as part of another form or another structure called chromatin. But what these chromosomes or chromatin contain? Both of them contain DNA and proteins only. And what is the difference between chromatin and chromosomes? Chromosomes are thick, condensed, rod-shaped structures, whereas chromatin is thin, entangled mass of thread-like structure. That is the difference, right? So the genetic material is actually DNA, which is wrapped around itself with the help of proteins, and it is in very thin form of structure called chromatin when the cell is not at its dividing stage, whereas when the cell is about to divide, it is going to be present in chromosome form, right? So chromatin material, it is visible as thin entangled mass of thread-like structure. Thin entangled mass of thread-like structure. And chromosomes are thick condensed rod-shaped structure. And what is the similarity in both of them? Both of them contain DNA and protein only. And what is the difference between them? Thick condensed rod-shaped structure is chromosome, which is present when the cell is about to divide. Thin entangled mass of thread-like structure is chromatin, which is going to be present when the cell is not at its dividing stage. Okay. Let me write about chromatin also. Thin entangled zigzag mass of thread-like structures. When the cell is not at its dividing stage. This zigzag structure is going to be present. Okay. When the cell is about to divide, what will it happen? The chromatin material will get organized. It gets organized into chromosomes. Why it gets organized into chromosomes? In order the genetic material to get split equally to the daughter cells. If it is in thin entangled mass of thread-like structure, splitting or dividing will become difficult. Not only that, separation of the functional segments into two equal components also becomes difficult. So only when the cell is about to divide, the chromatin material gets organized itself into chromosomes. What is the function of nucleus? Nucleus, it plays a central role in cellular reproduction. Cellular reproduction means a cell divides into daughter cells. In the case of normal growth and development, a cell divides into two daughter cells, which is generally called equational division. During the time of gametic formation, specialized cells, gamete mother cells, they undergo a reductional type of division in which the cell divides into four daughter cells. For this particular activity of cellular reproduction, dividing the cell into daughter cells. What is cellular reproduction? Cellular reproduction is the process of dividing the cell into daughter cells, as per which all cells arise from pre-existing cells. So nucleus plays a central role in cellular reproduction, and it also plays a crucial role in determining how the cell has to develop and what size and shape that it must exhibit by the time of full maturity. So nucleus also plays the role about the way of development of cell by the time of maturity and what type of form that the cell should get by the time of maturity, so that it must exhibit its function. These are the two important functions that are played by the nucleus. Okay, remaining aspects of nucleus we will discuss in the next class.